IB Bio, Cell Structure and Function Part 8 focuses on osmosis, the diffusion of water across a membrane. Osmosis is more complex than meets the eye because water is the solvent, not the solute, in biological solutions, such as cytoplasm. The diffusion of water across membranes is dependent on the concentration of solutes in the solution. The essential idea here is that membranes control the composition of cells by active and passive transport. Here's an outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the unit that you need to review. This movie is focused here. Here are our first two important IB syllabus statements of this movie. Define osmosis. Describe the circumstances by which water diffuses across a membrane. Remember, diffusion is defined as the net movement of particles from regions of high concentration to regions of low concentration. In this diagram, there are two solutions on either side of a membrane. The membrane, like all biological membranes, is semi-permeable. Some substances can pass, while others cannot. In this image, there are two different particles that could diffuse, the water and the sucrose. But the sucrose molecule is too large to pass the membrane. Thus, sucrose cannot diffuse, but water can. Now, water molecules, like all molecules above 0 degrees Kelvin, are in motion, and as such, water will move back and forth. But net diffusion of water will occur from left to right. Notice this pink arrow, from a region of high water to a region of low water. Osmosis occurs from the left solution to the right solution. Again, in this diagram, we can see osmosis as water diffuses from the left solution through a membrane to the right solution, from regions of high water to regions of low water. The sugar is the solute, while the water is the solvent. So another approach to the language when describing osmosis would be to say that water diffuses from low solute concentration to high solute concentration. When there is a membrane, water, the small particles here, move across the membrane more quickly than a solute. The solute may not be able to diffuse through the membrane at all because of size or charge. Osmosis occurs from regions of low solute, left in this image, to high solute, right in this image. So osmosis can be described using language in three different ways. It's the diffusion of water across a membrane from regions of high water to regions of low water. Or it's the diffusion of water from regions of low solute concentration to regions of high solute concentration. Or it's the diffusion of water from hypotonic solutions to hypertonic solutions. Now, this last statement uses language that I've not yet used in this movie. Let's look at these terms, hypotonic and hypertonic, more closely. Now remember that cell cytoplasm and the fluid outside the cell are solutions, composed of solvents within which solutes are dissolved. In biological circumstances, the solvent is always water. A solution with a low solute concentration, like this left beaker, is a hypotonic solution. A solution with a high solute concentration, like this right beaker, is a hypertonic solution. Now, what happens to cells when placed into solutions of different concentrations of solutes? In what direction will water diffuse across the membrane? Consider the terms that describe the relative concentrations, hypotonic, hypertonic, and isotonic, which means that the concentrations of the solute are the same. Case 1, a cell is placed into a hypotonic solution. The cell has a higher solute concentration than the beaker. The cell is hypertonic relative to the solution in the beaker. I'll use three different language patterns to describe what happens, to describe what you can see here. Water will diffuse from the high water of the beaker into the cell. Osmosis will occur from the low solute concentration of the beaker to the high solute concentration of the cell. Osmosis occurs from the hypotonic solution of the beaker to the hypertonic solution within the cell. The cell swells, and and if the cell is an animal cell without a wall, the cell could burst. Case 2, a cell is placed into a beaker that is hypertonic. The cell cytoplasm is hypotonic relative to the solution into which it's placed, which is hypertonic. You can see that water leaves the cell, moving from the hypotonic solution out to the hypertonic solution. The cell shrinks. It's a process called plasmolysis. Case 3, a cell is placed into an isotonic solution, a solution where the concentration of solutes is equal on both sides of the membrane relative to the cell. Thus, there is no net movement of water. No net movement of water. Now, water does move back and forth randomly across the membrane, but there is no net movement, thus no osmosis. When an animal cell is placed into a hypotonic solution, the diffusion of water into the cell causes the cell to swell and possibly burst. If a plant cell is placed into a hypotonic solution, the diffusion of water into the cell causes the cell to become turgid, rigid with the influx of water. When cells are placed into isotonic solutions, water randomly moves back and forth across the membrane, but there is no net movement, no osmosis, no change in cell size. When the cells are placed into hypertonic solutions, water diffuses out of the cell, the animal cell shrinks. In plant cells, the membrane pulls away from the cell wall. This is called plasmolysis. So all of this raises an interesting question, which is the subject of this IB syllabus statement. Briefly describe why tissues or organs to be used in medical procedures must be bathed in a solution with the same osmolarity as the cytoplasm. Donor organs are bathed in a saline solution that is isotonic to body fluids or cell cytoplasm. This prevents tissues from swelling or shrinking, which would reduce transplant success. Kidney dialysis uses membranes and diffusion gradients to adjust the composition of the blood that's withdrawn from the patient, exposing the blood to solutions of varying concentrations. Water can be added or withdrawn, and waste substances like urea can be withdrawn. In this image, you can see the high surface area of the kidney dialysis machine. This high surface area over which the blood flows allows the blood to be exposed to these various solutions that adjust the composition of the blood as it returns to the body. Here's a chance to practice your understanding of osmosis. In this diagram, we can see two solutions set up on either side of a membrane at time 0. And then at time 1, the solution on the right has increased its volume. Now turn the movie off now or follow along with me. At time 0, the solution on the left was hypotonic relative to the solution on the right. This is hypertonic. The solute concentration on the left side of the membrane was less than the solute concentration on the right side of the membrane. These solutes are too large to pass the membrane or too large to pass the membrane quickly. Water then diffuses from the hypotonic left side to the hypertonic right side until both sides are isotonic, but this results in an increase in the volume of water on the right side of the tube. When this thistle tube, containing a sucrose solution, was placed into distilled water with no solutes whatsoever, water diffused across the membrane, resulting in the rise of water measured as D. Depending on the concentration of the solution, water can be drawn toward a solution to a greater or lesser extent, as can be seen in this image. The degree to which a solution's concentration results in water diffusing into the solution is called osmotic potential. Here's another chance to practice your understanding of the direction of osmosis with the given concentration of solutions within the cell and in the beaker at the start of an experiment. What would happen over time? Now, you can turn the movie off now or follow along with me. Water will diffuse into this cell, because the solute concentration here is higher within the cell than it is outside the cell. The cell will swell, potentially burst, without a wall. Here's the next IB syllabus statement related to the diffusion of water. In what way do plants rely on high hydrostatic or turgor pressure in their cells? Explain how this condition occurs. In order for plants to remain upright, plants require water to diffuse into their cells. When water diffuses into plant cells, the cell becomes turgid, rigid. Plants rely on solutions that surround their cells to be hypotonic relative to the plant cell. If plants do not receive enough water, their cells lose water, the plasma membrane pulls away from the cell wall, the cell becomes plasmolized, and the plant wilts. The plant cells in this upper image have been placed in salt water, a hypertonic solution. Water diffuses out of the cells, and the cells become plasmolized. The plant cells in the lower image have been placed in distilled water, a hypotonic solution. Water diffuses into the plant cells, and the cells become turgid. Here's the last IB syllabus statement of the movie. Define water potential. Describe osmosis using the term water potential in your description. Water potential is particularly important in studying the movement of water into and out of plant cells because the cell cytoplasm has osmotic potential, but the cell wall pushes back against the influx of water, so there is pressure potential in the wall in addition to the osmotic potential of the cell cytoplasm. Water potential is a measure of the tendency of water to leave one location for another location, and we're dealing with plant cells here. Whether or not water will move into or out of a plant cell depends on both osmotic potential, a function of the solute concentration of the cytoplasm, but also pressure potential, a measure of the pressure that the cell wall applies to the cell cytoplasm. To follow in the pattern of defining diffusion, osmosis can be defined as the net movement of water across a membrane from the more positive water potential to the more negative water potential, from high water to low water. Here's water potential in its definition once again. Water potential is a measurement of the tendency of water to leave one location for another. Water potential is a function of the pressure potential of the wall and the solute potential or osmotic potential of the cell cytoplasm. Osmotic potential is a complicated measurement, but let's keep it simple and note that it is dependent on the concentration of the solution. And notice here that the more concentrated the solution, the more negative the osmotic potential. Now this should make sense. Hypertonic solutions, like these at this end of the table, would not tend to lose water, but instead have water diffuse toward them. So let's apply what we know to a problem. I think you'll see it's not as complicated as it might appear. Water potential is the sum of the osmotic potential of the cell cytoplasm and the pressure potential of the wall. The water potential of the solution in this beaker is negative 130 as the sum of the solute potential and the wall pressure. There is no pressure of wall in this open beaker. So the water potential of the solution is negative 130 kilopascals. The water potential of the cell, again, as a sum of the solute potential of the cytoplasm and the wall pressure, is negative 260, the sum of the solute potential and the wall pressure. The wall pressure is zero simply because there is no pressure of this wall on the membrane. The membrane is not in contact with the wall. Now, if we compare the water potential of the beaker, negative 130 kilopascals, with the water potential of the cell, negative 260, we will see that water will move from the more positive toward the more negative. Water will flow from the beaker into the cell. Water will flow from the more positive water potential to the more negative. Water will diffuse into the cell. So as the water enters the cell, the wall pressure begins to increase. The pressure of the wall pushes back on the arriving water, increasing the pressure potential And that changes the water potential of the cell. The point at which the water potential on each side of the membrane is the same as it is here, minus 130, if you sum these two values, and minus 130, water will no longer diffuse. Water no longer diffuses into the cell at this point, even though the cytoplasm of the cell is hypertonic relative to the solution in the beaker. The wall pressure prevents more water from entering the cell. Here's an easy question because neither the beaker nor the cell have a pressure potential. So just compare the osmotic potential to determine in which direction the water will diffuse. Water diffuses from the more positive potential to the more negative. Water will diffuse into the cell. Here we have two cells, side by side, separated by membrane, and I'm asking in which direction will water diffuse, osmose, over time. The left cell has a total water potential of -1 atmosphere, while the cell on the right has a water potential, total water potential of -2 atmospheres. Do you understand how I've calculated those water potentials on each cell? Water will diffuse from left to right, from the more positive potential to the more negative potential, from -1 toward -2. So here we have a plant cell that's placed into distilled water, which is very low solute concentration, very hypotonic. It's an open beaker, so there's no pressure potential. You can see that the membrane of the plant cell has pulled away from the wall slightly. Nevertheless, the wall is applying a +1 atmospheric pressure, while the solute potential of the cytoplasm is -4. This is at time 0, and we slide to time 1. You can see that the cell has taken on water. The wall pressure has changed. Wall pressure is now +3. And I'm asking, what's the solute potential of this particular cell? With water no longer entering, the solute potential would be -3 atmospheres. And that brings us to the end of IB, Cell Biology, Part 8, on to Part 9, the last movie of the series.